In this video, John MacMicking talks about his research on a family of immune proteins that release deadly sacs of antimicrobial peptides to kill bacteria. The paper will appear in the May 6th issue of Science.
The idea is that these GTPases are chaperones. They more or less reside on small vesicles. These vesicles or sacs contain noxious or nasty antimicrobial components that, as I said, are then delivered to where the bacteria reside. And so one of the antimicrobial proteins that these GTPases help deliver is an enzyme called phagoside oxidase, which in turn generates what's called a chemical or radical called superoxide, which eventually can be oxidized to hydrogen peroxide, which I think many of us know is often part of the bleach that we administer to the kitchen or the sink or wherever we find microbial flora growing in our own household. So our own cells have the ability to make chemicals that we, as I said, can normally manufacture and bottle into something like bleach, as I said, as a natural evolutionary way to control infection by bacteria or fungi or viruses or protozoa, for example. It's basically looking at an immune cell called a macrophage that's been activated with immune interferon gamma. The green dots or green vesicles, if I remember rightly, are in fact one of these GTPases called GBP7, and the red bacterium in this case is Mycobacterium bovis BCG. And so what you can see is that these this particular GTPase gets targeted onto the mycobacterial vacuole about 30 minutes after uptake of these bacteria into the infected macrophage. The reason we chose these two particular bacteria were there's quite a academic history and experimental and clinical history of humans, for example, that have mutations in the interferon gamma pathway. There are some rare mutations in humans in this pathway, either the receptor, the ligand, or adapter proteins downstream of the receptor. Those people often present at the hospital or the clinic with uncontrolled mycobacterial infections. They can be either tuberculosis or non-tuberculous mycobacteria. They often present with disseminated listeriosis, so they can't control listeria infections, or salmonellosis. They can't control salmonella infections. So we already have in the human population genetic knockouts, if you will, just like mouse knockouts that we've used in this paper, that tell us that we need the interferons for controlling certain types of bacterial infection. And of course, there are, as you say, many bacteria that we know are controlled by this particular immune hormone or immune cytokine in experimental systems. If you genetically remove it, mammalian hosts are highly susceptible to a variety of different bacterial species. So we anticipate that these proteins, as we go along, will fulfill important protective functions against bacteria besides these two that we happen to have studied.
This video was produced by the Office of Public Programs at AAAS, the nonprofit science society that publishes the Science family of journals.